Over 253 million children worldwide are estimated to be vitamin A deficient. To put this huge number into context, that's over 6 times as many people as the entire population of Canada. Although vitamin A deficiency, or VAD, is highly prevalent in regions where diets are nutritionally lacking, it remains an issue largely out of the public's mind. Vitamin A is a micronutrient that's essential for the growth and specialization of virtually every cell in our body, and VAD is not only a leading cause of preventable blindness, but it also increases risk of infection, disease, and death. Many mitigative initiatives have been trialed, but these solutions have experienced limited success over the long term. A prominent example is vitamin A supplementation programs, which require a government or foreign body to provide supplements, meaning that any disruption to the supply chain can put communities right back into a vitamin A deficit. Another prominent example is the Golden Rice Project, which has experienced limited implementation over the past 2 decades for a number of reasons, including its extensive growth requirements and poor biocontainment practices. So, if we wish to see vitamin A deficiency successfully mitigated, deficient communities need a solution that can be sustained over time. More importantly, they need a solution that they themselves can sustain over time. My name is Simran, and along with my colleagues Namaya and Randy, we're going to be telling you about how our solution, Ovita, equips vitamin A deficient communities with a sustainable and community-based source of vitamin A. To start, Ovita entails a nutritional yeast rich in vitamin A and healthy fats that can be sustainably cultivated by vitamin A deficient communities as an edible supplement. But before I get into any more details, let's go back to the very beginning with the regions that are affected by VAD. We knew that VAD is most prevalent in South Asia and Sub-Saharan Africa, but from conversations with our HP contacts, we decided to focus our initial implementation efforts on regions such as Ghana, where pre-existing community-based brewing infrastructure could be adapted for the cultivation of our yeast. We also knew from our conversations with anthropologists and field experts that people in these regions are generally accepting of GMO products provided they're of benefit to them. And we're currently in the process of reaching out to overseas community contacts to gain further insight. Once we knew our VAD regions, our next step was to choose a yeast strain that satisfied the following criteria. First, our choice of yeast had to be capable of growing in hot, dry climates. Second, it needed to be capable of producing vitamin A. But since vitamin A itself can be toxic in high quantities, we decided to eliminate the risk to our consumers by finding a yeast capable of producing beta-carotene, a non-toxic precursor that can be naturally converted by the body into vitamin A. As vitamin A is a fat-soluble micronutrient, the body requires lipids for successful uptake. To ensure its effectiveness as a supplement, we therefore decided to choose a yeast strain that's oleaginous, or rich in lipids. And since our supplement was going to be ingested, our fourth criteria was recognition as food safe. Now, with these 4 criteria in mind, we still needed to ensure our yeast could be sustainably and simply cultivated by VAD communities. So we met with Dr. Charles Mather, who informed us that individuals in our target region frequently reuse agricultural waste for a variety of purposes. From this, we were inspired to use agricultural waste, which is rich in cellulose, as feedstock for our yeast. In order to do this, we needed a yeast that was capable of being modified for cellulase production. As cellulases are a set of enzymes that convert cellulose into glucose, this modification would enable our yeast to grow using cellulose-based agricultural materials, such as rice husks. After the careful consideration of many organisms, we selected Uroya lipolytica. As an oleaginous yeast, Y. lipolytica can tolerate dry and hot climates and is generally recognized as safe. Furthermore, past strains of Y. lipolytica have been engineered to produce beta-carotene and cellulases, although no strain has been engineered to produce both simultaneously. We therefore set to work on engineering Y. lipolytica for simultaneous beta-carotene and cellulase production. Fortunately, we were able to obtain a strain of Y. lipolytica that's already been modified for beta-carotene production. So all we have to do is engineer Y. lipolytica to produce cellulases. To do this, we'll introduce 3 types of cellulase genes into the genome to allow for the secretion of the enzymes to the extracellular medium. Once the enzymes are secreted, they can work together to break down the cellulose. In an effort to reduce the strain placed on the growth of the organism, the 3 cellulase genes will each be integrated into different yeast strains. Thus, the different yeast cells will need to be co-cultured to allow for the successful degradation of cellulose to glucose. For this effective degradation, we require 3 types of cellulase enzymes: endoglucanase, cellobiohydrolase, and beta-glucosidase. There are 2 endoglucanases required, 2 cellobiohydrolases, and 1 beta-glucosidase, making a total of 5 enzymes, each with a specific role to play. However, we wanted to optimize our cellulase enzymes to work best in Y. lipolytica. So now I'll hand it off to Namaya to explain how we did that.
Using molecular dynamics, we modified our enzymes to be suitable for the conditions that we want. Luckily, cellulases have previously been heavily characterized, allowing us to take advantage of a number of mutations that can have a strong impact on thermostability and optimal pH. Ultimately, through our modeling, we were able to produce optimized versions of the CBH1 and EG1 enzymes, where the optimal pH of each protein was increased by 1 unit. Additionally, we were able to reduce the ideal temperature required for the functioning of the CBH1 enzyme by 2 degrees. Now, these changes are significant because these newly modified enzymes now allow us to improve the functioning of the cellulases to the conditions expected in our system. So once we had optimized our cellulase enzymes to use in our system, we then set ourselves to create the genetic constructs. Each construct included a strong promoter, secretion tag, affinity tag, the cellulase gene, and the terminator. Since our plan is to have a different yeast strain secrete the 3 different cellulases, we designed 3 different constructs with a different affinity tag and a different cellulase gene. The different constructs would include either the 2 endoglucanases, the 2 cellobiohydrolases, or just the beta-glucosidase. We plan to assemble our transcriptional units using Gibson assembly and have also designed the constructs to allow for the future direction of including all 5 cellulase genes in one construct to be integrated into a single yeast strain. Our yeast, Uroya lipolytica, primarily uses non-homologous end joining for DNA repair, so we will be electroporating the linearized constructs into the yeast, allowing for the random integration of the constructs into the yeast genome. So once we actually get into the lab, assemble our constructs, and integrate them, we then plan to confirm the expression of our cellulase constructs and subsequent secretion of the enzymes using Western blot analysis. If we are successful in secreting the enzymes, we should see bands at the appropriate molecular weights in the extracellular samples. As well, we will be using a Bradford assay to quantify the concentration of proteins produced. And then, we will be making sure the cellulases are working together as expected and breaking down the cellulose to glucose. For this, we will be using a DNS assay to test for an increase in reducing sugars in the media. Since glucose is a reducing sugar, we can use this assay to confirm the successful degradation of the cellulose to glucose. We will also be conducting a glucose test to measure the exact concentration of glucose produced by the yeast. And finally, we will conduct co-culturing experiments with all 3 strains present to determine the growth of our yeast strains. And now, I will be handing it off to Randy to take you through how exactly we see our yeast growing in these communities.
We knew from the beginning of our design process that we wanted our bioreactor to be centered on the community that it's meant for. We knew that it should be simple, and to achieve this simplicity, we knew that our bioreactor should be instructed using locally sourced and low-cost materials. To work towards a sustainable solution, we also wanted to ensure that our bioreactor could operate off of agricultural waste. Chatting again with Charles Mather, who has in fact worked in northern Ghana's communities, he brought to light an interesting point. Ghana has an active beer brewing community where they use terracotta pots to brew for the community. Now, obviously we don't plan on making beer for children, but we thought that utilizing the local knowledge and infrastructure on this brewing process would make Ovida much more realistic. Given this information, we began work on our bioreactor design. And drawing inspiration from the beer brewing process, our bioreactor came to look something like this: a large vessel with an air pump and a source for electricity. But this was no good. All we had were some soft constraints and a vague idea on components. Constructing models on the growth of Uroya lipolytica, we were able to better understand the needs of this organism and define hard constraints on our design. Not only do these elucidate the conditions needed for optimal growth, They also verified our design, and preliminary models indicate that a satisfactory amount of beta-carotene can be produced in a matter of days. Factoring these numbers into our design, we were able to get rough dimensions to our bioreactor. We now intend to iteratively refine our models and bioreactor design by constructing a laboratory bioreactor to get experimental data on our specific strain. But designing the bioreactor was only half the battle. To make our solution effective, we had to make it something that people would actually eat. Luckily, the European Food and Safety Authority deemed the wild type of Uriella lipolytica to be food safe as long as it is heat killed and dried. Although a genetically modified organism would have to go through rigorous testing to ensure that it too is food safe, the reason that heat killing will be the most probable way to prepare the yeast. Now, Uriella is assumed to have a meaty or cheesy flavor. Luckily, we were able to contact an cGEM team from Ghana to get a better understanding of local taste palates and see exactly where this yeast could fit in. Our own research found that curries, stews, vegetable and rice dishes were all possible ways to incorporate the yeast. We intend on continuing collaboration with teams to create our own cookbook. Now, investigating local foods, we came across another factor contributing to vitamin A deficiency. Namaya will now tell you more about this.
Even with a product to supplement the diet, something that we learned from another one of our HP contacts, Dr. Diasanu, is that diet isn't everything when it comes to combating vitamin A deficiency. In fact, intestinal parasites are highly prevalent in the communities we are targeting and play a key role in preventing the successful uptake of vitamin A by perforating the intestines. Organizations like the UN, World Health Organization, and the Bill Gates Foundation do exist to provide biannual deworming agents, but a lack of clean water and proper footwear still allow these parasites to return and thrive in these communities. Based on these issues, we plan to have our yeast engineered to produce thymol, an anthelmintic agent, a substance that will kill the parasites in the body. Thymol is a compound naturally occurring in thyme leaves and has long been used for its antioxidant and anti-inflammatory properties. It is proven to work against worms and their larvae, thus allowing the intestines to recover and maximize absorption of vitamin A in the body. Moreover, thymol is generally recognized as safe by the US FDA and may be ingested with negligible toxicity, making this compound an attractive solution to combating parasitic infections. Since lipolytica already has the precursors for the thymol biosynthesis pathway, when creating our constructs, we only have to engineer 2 genes into the genome, a terpene synthase and a cytochrome P450. Now, once again, when we get back into the lab, we plan to test the expression of thymol by our yeast. First, we will confirm the expression of our enzymes using Western blot analysis. Then, gas chromatography and mass spec will be used to determine if thymol is produced. And finally, we will be conducting some tests using C. elegans. C. elegans is the gold standard used for testing anthelmintic drugs. To prove thymol's activity, we will be testing its effects on C. elegans by soaking E. coli in varying concentrations of thymol and feeding it to the C. elegans before observing their growth. But with all of these modifications to our yeast, we also have to be mindful with safety, the safety of our users and greater environment. Though we designed our bioreactor to tightly contain the biological components inside, there is always the possibility for the escape of our yeast. For this possibility, we came prepared with a biocontainment strategy. Currently, one of the main strategies to mitigate safety concerns is through auxotrophy. Essentially, this means preventing an organism from producing a vital molecule that it needs to survive. As you can see in this diagram, these strains are auxotrophic for tryptophan, and thus require the external addition of tryptophan in order to survive. Unfortunately, such a strategy is not very useful for the long term. It introduces high costs and is ultimately unsustainable due to the external supplementation of the required molecule. So with that in mind, we designed our own biocontainment strategy that builds upon the principles of auxotrophy, but with a little twist. Our system relies upon the co-culturing of 2 complementary strains, wherein one strain is auxotrophic for a molecule that the second strain will overproduce. Essentially, we are forcing the strains to become codependent upon each other, The organism will only survive if its partner strain is present in high enough quantities to provide the molecule of interest. The overproduction of the molecule will be achieved through the integration of a gene that is resistant to feedback inhibition or through the introduction of a gene under a strong constitutive promoter. This season specifically, we will be focusing our efforts on testing a proof of concept for a biocontainment system, using a pair of already engineered Saccharomyces cerevisiae strains. These strains were engineered to be oxytrophic and overproduce an amino acid, as shown in the diagram. Now, with these strains already engineered, when we get into the lab, we will get right to conducting experiments to characterize the effectiveness of our system as a biocontainment strategy. First, we plan to confirm the oxytrophic nature of our strains and the ability of our cells to overproduce these amino acids. We will then determine the growth rate of our yeast when they are co-cultured together by measuring fluorescence and optical density. And finally, we will be testing the longevity of the co-cultures by determining the minimum cell numbers required to sustain a co-culture for an extended period of time. Ultimately, once we gain an idea about the functioning of our biocontainment strategy, next year, we plan to engineer Myelopolitica strains to give it these same capabilities. I will now be handing it off to Randy to walk you through how we used metabolic flux analysis to understand how our yeast was able to withstand all of these genetic modifications that we implemented.
What we really wanted to know was just how well our yeast functions given these genetic modifications. Without the lab, we decided to use metabolic flux analysis. Using Python to test a genomic scale model, we were able to observe changes in flux correlating to genetic modifications. We intend to further use this method to help identify how well our yeast can perform in bioreactor conditions. So, with an organism, a bioreactor, and models to guide us, we began to look for specific communities we could partner with. But this proved to be harder than we imagined. There was an inherent lack of community-specific data on vitamin A. Further talk with researchers and not-for-profit organizations made it apparent that decision makers needed more data to identify vitamin A deficient populations. We decided to tackle this problem head-on by creating a point-of-care diagnostic test for vitamin A deficiency. Our intended solution is a blood-based test that uses retinol binding protein as a biomarker for vitamin A deficiency. After separation of blood plasma from a sample, our test takes advantage of the selectivity of aptamers. The retinol binding protein from the sample selectively binds to an aptamer-coated electrode, causing a measurable change in impedance, which can tell us about vitamin A deficiency. Essentially, our device functions like an electrochemical cell. We've designed a circuit that uses impedance spectroscopy to measure the impedance change caused by the presence of vitamin A. The benefits of this method are that there's no laboratory analysis required, and it offers rapid testing. So from selecting bioreactor designs to understanding local practices, we wanted to see Ovida come to life, and we knew that to do that, entrepreneurship was key. The immediate challenges that we faced were the distribution of our product, the funding of Ovida, and the empowerment of those who will be operating our Ovida system. We turned towards partnerships as a way to tackle the monumental task of funding and distribution. We've made steps to create partnerships with other not-for-profits by incorporating Ovida itself as a not-for-profit. An advantage of Ovida is that our yeast, being a nutritional supplement, has monetary value, which encourages production. By supplying the tools in the form of Ovida, we hope to inspire micro-enterprises and empower rural entrepreneurship. Because community engagement is the spark that is needed to tackle vitamin A deficiency. And Ovida is a tool by which that spark can be ignited. Ovida is a solution sustained by the trust and imagination of the communities that it aims to support. It is a solution to vitamin A deficiency that is safe, sustainable, and community-based. Ovida is cGEM Calgary 2020. We would now like to take this time to thank our sponsors, as well as our countless professional and industry contacts. We could not have pursued this work without your support. Thank you.